Thank you, and thanks for the opportunity to come here and introduce Universal Cells to you. We're a small company in Seattle. Company's been in existence for roughly 5 years, or for 3 years, and we've made significant progress on trying to make stem cells compatible with everyone. If you think about what we've heard about today, we've heard a lot of discussion about the HLA matching strategies or autologous therapies. And I call that the hamster wheel, where we have patient stem cell isolation. You go into a processing to isolate the cells. The cells are variable because they each come from different patients. You have variable input results resulting in differentiation complexity. You then transplant and go back to the patient. Wouldn't it be nice if we could have a situation as shown on the left, where you start with a non-immunogenic pluripotent master cell bank. From there, you differentiate to the tissue type that you desire, and then you treat the patients with an off-the-shelf therapy. The benefits would be consistent raw materials, scalable manufacturing, off-the-shelf products, reduced cost of goods manufactured, simpler trial designs because you're not dealing with the patient-to-patient variability in your manufacturing, no immunosuppressive drugs, and hopefully consistent outcomes. As an old guy in this industry who's been doing process development forever in the biotech world, I like a linear process like this. I don't like circles and hamster wheels. When you think about it, what this allows you to do, if you think of the iPS or ES cell and the gene editing, this is very similar to the master cell bank creation where you make research stocks. At this particular time, you can do many things to the cell if you have the capability to do precision gene editing, such as HLA replacement, the introduction of suicide genes to improve the fidelity of the system and safety, genetic stability, master and working cell banking, robust monitoring or repair of oncogenes that might come out during the growth of the iPS or ES cells, as was recently shown in Egan's paper. And finally, adventitious agent testing and control using a paradigm that has been widely accepted for monoclonal antibodies and vaccine industry, which are used to treat normal, healthy individuals. And the differentiation process is actually very similar to the drug substance development process. Here you're working on how do I differentiate cells? How do I remove, through a purification process, undifferentiated cells? Because as we know, injecting a pluripotent stem cell could result in a teratoma. That is something you would not want to do, so you need to set a high bar for the fidelity of your conversion of cells to the desired target site. You can also think about what is the differentiated cell's stability at this point? Is it something that I can freeze? If I can freeze, then I truly could have off-the-shelf therapies to offer to patients. If I can't freeze, then I use a cell bank of the pluripotent stem cell that's been edited, and I run the manufacturing process in a just-in-time manner. So both options exist. You can consider scale up in a traditional volume sense, like pharma is used to, or you could do a number up options if you have to have rapid production of differentiated cells at the point of use. You then move to this third area, which to me is very much like normal product development. You're thinking about the convenience now. How will this product be used in the field? Delivery, devices, integrity, packaging, freeze-thaw, logistics, all of those types of things that are normally thought about and have been handled in pharma. Finally, clinical practice needs to define efficacy and, very important, training and compliance practices that are used by the physicians to actually get the patient. So with such obvious benefits, why hasn't it happened? Well, we know from years of transplantation research that polymorphic HLA molecules activate T cells and are responsible for a lot of the rejection that's observed in the clinic. All somatic cells have class 1 HLA molecules, and they come in the forms of A, B, C, E, F, or G, and there are polymorphs within each of those classes. The class 2 are presented on antigen-presenting cells, and are less important than class A, but depending on the tissue type that you intend to differentiate to, could be important for your application. The presence of the HLA mismatches then results in T cell activation and rejection. It would be simple to just say, let's eliminate all of the HLA molecules. We have gene editing tools that allow us to knock things out relatively easily, but it's not that simple. Your body actually contains natural killer cells who are designed to scan for rogue stem cells. And if you don't have markers on the surface, those natural killer cells will then lyse the null HLA cell. So our approach was to take an engineering approach and to say, for the class 1 molecules, we're going to remove them, but we're going to replace them with a structure that contains beta-2 macroglobulin linked to HLA-E. HLA-E was selected because it has 2 polymorphs and they differ between 1 amino acid. For the class 2, we can completely remove all those because there are patients who are perfectly happy without class 2 molecules on their surfaces. In order to do that, you need to have a very precise editing tool. We actually use an AAV-based system. AAV was selected because, one, it's already been used in clinical settings, and so there's a fairly well-known body of literature around AAV. Second, it's a very simple system to deliver to these stem cells. It's done on small scale, so we don't have to worry about the scale-up of the AAV virus. But most importantly, it's a nuclease-free technology. We do not have double-strand cuts. We have no genotoxicity, no off-target cutting, and no air-prone repair. That sounds like a perfect tool. Unfortunately, it is not as efficient as other tools, and so we have to screen many more clones to find those that are edited in the very precise manner that we would like. However, we believe that the increase in precision and the lack of off-target damage is worth the trade-off of doing a little more work up front. The mechanism of this is through homologous recombination. It's shown on the left-hand panel there, and I can go into that in detail if somebody would like to visit later. So we've made a research-grade universal donor cell. We started with parental cells that had HLA class 1, HLA class 2, and a small amount of HLA-E. This is a cartoon. We knocked out HLA class A and added back in the HLA-E that I described in the center panel. We also said, you know, if I'm going to make a universal donor cell that has the potential to cause teratomas and other unknown effects, why not introduce a suicide gene? And so we also introduced thymidine kinase. In the second step, then, we took out the HLA class 2 molecules by editing the RIF-SANK promoter region. It's not vaporware. Proof of concept was recently published in Nature Biotechnology, and I could not do justice to the science that's in this paper. I joined the company 3 months ago. David Russell is the Chief Scientific Officer. This is work out of his laboratory at the University of Washington. It's a very compelling story showing that we have, by editing the HLA molecules, reduced or eliminated rejection. Now, if you think about that, that creates a superior starting point for the development of a therapeutic product. Feasibility, we've shown that it is feasible. We actually have several partners that have already come on board who have asked us to edit cells on their behalf. Removal of the HLA and class 1 and class 2 molecules and introducing the single chain HLA-E and suicide gene, quite robust and easy for us to do. The cells, importantly, are karyotypically and behaviorally normal and retain their differentiation capabilities, at least in those systems that have been tested to date. In that paper, the Nature Biotech paper, there is no immune response in vitro or in vivo models. I should note these are animal models, so that the real test is when we put them into humans, and that's what the next stage of the company's development will be. But in those models, there was no stimulation of PBMCs, no T cell response, not targeted by allogeneic T cells, no lysis by natural killer cells, and it was not recognized by a panel of anti-HLA molecules in FACS-based assays. I contend it's actually a better platform if you're thinking of a differentiation approach. You have no genotoxicity, no off-target effects from the genome editing. We've introduced suicide gene to improve the safety profile. We use precision editing that's aligned with your target product profile to allow you to introduce characteristics that you might find beneficial. GMP banking with extensive adventitious agent testing, and no immune suppression, donor matching, or rejection. What we need are partners who have the differentiation processes down and who have the great ideas of how to use a fully differentiated stem cell to treat therapeutic needs. We have many collaborations currently underway. I find it ironic that the keratinocytes is the only class that we don't have at least an academic partner involved. All of those listed in green are currently at either academic or out-licensed, both in either exclusive or non-exclusive arrangements. Two of them have been announced publicly: adoptive immunotherapies. Adaptimmune has T cells, and the retinal pigmentosis, the RPE, has been licensed to Helios. It's important to understand how we are approaching this. So, gene editing is not a deterministic process. Producing the universal donor cell is not a GMP process. It is very analogous to producing a cell bank used in the monoclonal antibody field. We go through multiple cycles of gene editing and clone selection to create these. Fortunately, the stem cells are immortal. Immortal stem cells allow you to create banks and to expand them almost in an unlimited fashion. At Universal Cells, we have made the conscious decision to keep the gene editing as a research process, but we have supplemented it with extensive documentation and raw material controls. We routinely evaluate starting materials, which in this case are the iPS or ES cells, thinking of donor consent, donor testing, patient histories. We have raw material documentation and risk assessments associated with them, written procedures, quality controls, and in-process criteria. And certainly we can verify the gene editing by PCR flow, Southern, or other sequencing techniques like whole genome sequencing. Finally, it's our practice to convert the research stock to a GMP master cell bank, doing that in a GMP facility under a fully GMP documentation setup. We have freedom to operate. Our IP position gives us the freedom to both operate and can block others from similar approaches. There are 5 patent families associated with the work that I've described here. We are welcome partners who want to take advantage of this platform. We believe it is a superior platform, and we believe that we offer a great value proposition. It's a very small company. At the top we have Claudia Mitchell, who's a CEO, comes from the regenerative medicine. David Russell is the Chief Scientific Officer out of the University of Washington. I came to the company 3 months ago to bring in process development, manufacturing, quality, and regulatory. And if you're interested, contact Gregory Block. He's in business development and would love to hear from you. Dedicated facilities right now are 5,000 square feet. Those will actually grow to 10,000 feet before the end of the year. We are 19 employees, and as was mentioned before, we are hiring as rapidly as we can identify highly talented and motivated scientists. It's not a bad place to live. In Seattle, we are right close to the Space Needle, and on rare occasion, you can see all the way across to Mount Rainier. Thank you.